It's just like a lot easier. You don't have to deal with everybody logging in and everything like that to Zoom, like especially like for family
but
and
it's
stuff.
more passive on their.
Yeah.
Okay.
Yes.
Okay.
Okay. So, all right. No, I am trying to get the YouTube stream to work. Okay, there I am. Copy. Okay. All right, and now I'm gonna send it to you.
Thank you.
Yep. Hi, Dr. Lyles.
Full representation of the Stuart's going to join us.
Okay. I'm getting there. I should.
Yeah, people are popping off.
Yes. Okay. Hi. Oh, you can see me texting. It's like a 30-second delay.
So
Okay.
you have it going?
Awesome.
Where's the link?
Yeah, I just emailed it to you.
Okay.
Yes, I put it in the group chat. Hi, everyone. And Dr. Price is on there as well.
All right.
Okay, a little bit of technical difficulty, but we are there.
Okay.
Thank you all.
Yeah, so,
They're there later, sorry, they're really there. They're really there. I didn't get enough for every layer. The committee will be well fed.
no false advertising.
Okay. Do you know if so it's working?
Yes. Yep.
Okay, so the attendance should go up from
Yes.
DES.
Yeah, there's 12 watching currently.
Okay.
So we are getting there. Yeah, Laura is here. Dr. Price is there. Yeah.
Okay.
Cool.
Are you ready to go?
Yeah,
Okay.
ready as I will be.
So for all you faculty in attendance, either online or in person, please fill out the evaluation form of Haley's presentation this morning. And thank you all for joining us today. I'm really, really proud today. Haley joined the lab in 2015. So maybe I've been hanging on to Haley a little too long. She has done a fantastic job and has had projects that ranged from the cellular immunology all the way to computational analysis of microbiome. So it's been quite a journey and has settled on some really interesting data, both in vivo and in vitro. So just a little background on just who this Haley Hallowell is. She just fell out of the sky by emailing me in, I guess, 2014 from Gannon University in Erie, Pennsylvania. And that doesn't happen that often here. I mean, I love our students, but we're really good at recruiting sort of locally, right? So to have someone from so far away just sort of contact me and say, I'm interested, and then come down, have an interview, fit in really well with the lab group, get their thumbs up, and join us right away was like, hey, this is great. And every minute that she's been here has been just a joy. So she's had quite a role not only in the lab but in the department. She has been just about every officer in the student government, the Graduate Student Government Association. She's been the instructor of record for two really important courses, the Biology 1020, a large course, as well as Cell Biology Lab, which is a substantial achievement for a graduate student to have done that much teaching and do it really well. And she's won awards for just about every research conference she attends. She brings home research, either for presentations or posters, she brings home awards. And there was, we call it, the lab is very good at giving, and I should mention Haley is the last of a cohort of students that really had quite its own personality. And I'm very sorry to lose Haley because there's a lot of institutional memory that goes there. But Haley became known in our lab as the IT department. And that's because she's really, really amazing at various forms of computation, all of which remain mysterious to me. So she has developed a pipeline for the analysis of microbiome data, which has really helped us move forward and achieve, be able to publish the data and analyze it in a more meaningful way. So with today, she's going to tell you about two really interesting projects that she's worked on, and they sort of together lead us to some new questions that hopefully will be carried out in a sort of combined in vitro, in vivo system. All right, Haley.
Awesome. Thank you so much, Dr. Schwartz, and thank you all for coming to my defense today, which is titled The Phage Behind the Curtain, Determining the Role of Phage in the Restructuring of Gut Microbiomes During Disease. And if you look behind my title, you will notice the yellow brick road leading up to the Emerald City and the Wizard of Oz. And much like the wizard is the man behind the curtain, I think of phage as kind of the man behind the curtain in the gut microbiome, where not many people pay attention to them, but they're really kind of running the show in terms of influencing the form and function of the microbiome during the development of disease, and that's what I hope to convince you guys of today. So microbes and more generally microbiomes are everywhere on the Earth's surface. They're found in more common environments such as soils and oceans, and also in more extreme environments such as the hot springs in Yellowstone and deep sea hydrothermal vents. And they are so ubiquitous on our planet that it wasn't until 2019 that we actually found a place that was devoid of life, which is the Dalal acid lakes in Ethiopia. So it has to be a very extreme environment for microbes not to exist. But more specifically, I am interested in host-associated microbiome. So if we take a look, say, at the human body, there are various places on us and within us that have their own unique microbiome associated with it. And even more specifically, I am interested in the intestinal microbiome, which is a diverse and dynamic microbial ecosystem harbored within our intestine. And it contains all four domains of life. And over here, I just kind of have a simplified depiction of the microbiome. So we have microbes from bacteria, eukaryotes such as fungi, archaea, and viruses such as bacteriophage. And the gut microbiome is required for the stable and healthy function of multiple organ systems within the host. And the composition is determined by a variety of factors such as diet, age, antibiotic usage, and disease state. So there's a lot of evidence for kind of a causal relationship between the microbiome and disease. Two of the most widely studied are IBD and obesity. So we have this impact of disease state on the gut microbiome that kind of shapes its form and function, and in turn, the microbiome can then contribute to the disease state and its progression. But a lot of what we know about how disease states impact the microbiome is solely based on bacteria. Much less is known about how other constituents of the microbiome, such as phage, are impacted. So I'm going to take just a quick detour and give you guys a little primer about bacteriophage. So they are prokaryotic viruses that infect bacteria, and over here on the right, I have a little depiction of just some diversity that we can see in phage. This is not representative of everything, but it's a good kind of cover of it. And most what we find in the gut belongs to the order Caudovirales here, which are double-stranded DNA tailed phages, belonging to Myoviridae, Podoviridae, and Cephuviridae. And generally, they can be classified based on their life cycle or their reproductive cycle. They can be classified as virulent or temperate. So I have just a little figure over here kind of explaining that. So if we start with free virions, if we look at virulent bacteriophage, they are strictly lytic, meaning we can have a virulent phage attached to its host, adsorb to it, and inject its genetic material. It will undergo replication of its genome, translation and transcription of its proteins. It will subsequently assemble and be released from the host. And it's kind of this linear pattern with virulent phage. However, with temperate phage, it can undergo this cycle, but it can also perform what we know as lysogeny. So after this initial step of injecting its genetic material into the host, it can subsequently integrate into the host genome or exist as an extra chromosomal element and kind of hang out there in waiting. And it can be passed along many generations of bacteria, and not until we have a prophage induction event, and I should say when it is integrated into its host, it's known as a prophage. So not until we have this prophage induction event do we see the temperate phage kind of continue on into the lytic cycle and subsequently produce virions. Bacteriophage are the most abundant agent on Earth. There's estimated to be about 10 to the 31st phage on the planet, and most often in their given environment with their hosts, they're at a 1-to-1 or 10-to-1 ratio with them. So unlike bacteria, there has not been a defined core of bacteriophage found in the gut. There was kind of a landmark study that came out of Ruth Lay's lab recently where they took a ton of human subjects and looked at their virome over time. And what they found was that there was a lot of inter-individual variation between subjects, but that unique consortia of their virome, or their collection of viruses within their gut, was stable over time. And there has been some work looking at how bacteriophage populations are influenced by disease state, and specifically here I'm talking about IBD. There was a landmark study out of Skip Virgin's lab in 2015 where he looked at patients with colitis, and not only did he see kind of an expansion and an overall increase in bacteriophage number in the gut, but he saw that those populations changed in abundance. And that kind of led me to think, well, if we see phage change during IBD, would this happen in obesity? And you might be thinking, Haley, IBD and obesity are very different diseases. And if we're looking at clinical presentation, you would be correct. But at their core, they are inflammatory diseases. So IBD, we have these really acute, strong bouts of inflammation, while in obesity, we see this kind of long, chronic, low-grade inflammation develop over time. So with the first part of my dissertation, I was really interested in answering this kind of black box here. So does obesity lead to changes in bacteriophage populations? And second, because we see this kind of time component in the development of low-grade inflammation over time, I was interested in what role time was playing kind of in this model. And looking kind of holistically at this through metagenomic sequencing, it can give us a good idea of how community structure changes over time, but it doesn't really tell us about a mechanism, right? So for the second part of my dissertation, I was really interested in looking at what mechanism might be driving changes in bacteriophage populations. So say here's kind of our initial microbiome, our little consortia. Over here we have, say, our obese consortia, and we have these big changes in viral populations. So I was really interested in kind of answering this black box here. And there are many different things that could kind of influence changes in viral populations, such as immune activation, nutrient composition, or other intrinsic or extrinsic factors. So overall, the major goal of my dissertation was to understand how disease-induced intestinal inflammation influences bacteriophage host dynamics in the gut microbiome. And within this, I had two major project goals. First, with my first results chapter, I was interested in determining how bacteria and phage populations change in response to the development of obesity. And second, I was interested in how immune activation might contribute to alterations in phage populations during intestinal inflammation. All right, so we'll get into my first chapter, and I'm sorry this is a long title, which is titled Longitudinal Analysis of the Intestinal Microbiome in Obese Mangalitsa Pigs Reveals Alterations in Bacteria and Phage Populations Associated with Changes in Body Composition and Diet. And this is published in Frontiers in Cellular Infection and Microbiology. So one of the most important things that microbiome research have to do when we're setting up an experiment is kind of decide what model we're going to use. So most often we use humans or mouse models to look at the relationship between microbiomes and disease. But lots of issues come with these, right? So I couldn't take Dr. Cobine, put him in a sterile room, feed him sterile food, and collect his poop. Like that, that just wouldn't work, right?
You might be up for that.
Yeah, you might be up for that, but probably wouldn't pass eye. And there's lots of issues when it comes to mouse models, as they do not develop obesity readily without genetic manipulation. So luckily, when I first started in the lab, we kind of fell into a really great collaboration with a professor in agriculture that allowed us to use a model that kind of married the best of both worlds between these two models. So I'd like to introduce you guys to the very cute and woolly Mangalitsa pigs. So these pigs are a swine breed that is native to Hungary, and they are naturally obese due to a leptin resistance. So leptin is our fullness hormone. So when you leave this and you eat breakfast, you will release leptin that will signal to your brain that you are full and that you should stop eating. Mangalitsa pigs cannot detect leptin, so they never get that fullness signal, and they will just eat and eat and eat until they eat themselves into an obese state. And to date, this is the first natural, non-human model of obesity, and when I say natural, I mean not genetically manipulated. So another really cool thing about the Mangalitsa pig is nobody has ever characterized them before in terms of an obese model or what their microbiome looks like. So before I could really dive into the questions that I was interested in, we had to determine kind of the endpoint of a obese Mangalitsa pig and a lean Mangalitsa pig. So my first question was looking at how the microbiome compositions differ between a lean and an obese Mangalitsa pig. So to do this, we had two groups of 5 pigs each. We had our ad libitum group over here on the right, which was given unlimited access to a basal pig feed. And then on the right, or on the left here, we had our limit-fed group, which was given 40% of the caloric intake of the ad libitum groups. So after they were on the diet for a little bit over a year, our collaborators collected a bunch of phenotypic and metabolic parameters to kind of characterize them as a model of obesity, and our lab collected feces for metagenomic sequencing. So after we collected the poop, we extracted DNA, and it was sent off to undergo shotgun metagenomic sequencing on an Illumina HiSeq. And when we got those reads back, I ran them through the in-house annotation pipeline I developed. And if you have any questions about that, please ask me at the end. And from my annotations, I was able to perform a differential abundance analysis using DESeq2 to look at what populations were differentially either differentially increase or decrease in our different groups. I performed a Bray-Curtis dissimilarity index to look at kind of the differences between our samples holistically, as well as a Pearson's correlation to determine the relationship between microbial populations and metabolic parameters. So let's get into some of the metabolic characteristics of our endpoint pigs. So as you would expect, our ad libitum animals gain significantly more weight. And when we looked to see where that weight was coming from, either subcutaneous fat or muscle deposition, it really was driven by this increased deposition in fat. And I believe at the very end, when we took these measurements, these pigs were about 70% lipids in body mass. So they were very, very, very fatty. And when our collaborators took adipose tissue and did some mRNA expression experiments, they found that the ad libitum groups had a significant increase in adiponectin and steroid-CoA desaturase, which are an adipogenic hormone and protein, as well as an increase in leptin, which is kind of pointing to that leptin resistance, as well as an increase in PPAR gamma, which is kind of an inflammatory marker. So all of this stuff is kind of lining up towards what we see during human obesity. And then we took a look at their glucose and insulin levels. So not only were they glucose intolerant, seen here by them taking kind of longer to clear glucose from their blood, but they were insulin resistant. Again, pointing to kind of validating this as a model of human obesity. So let's get into the good stuff. Let's take a look at their microbiome. And so just to kind of orient you, I will typically have a little bit of a legend over here pointing to the major differences I want you guys to kind of pay attention to, because I know this data can kind of get a little messy. So this is relative abundance of bacterial phyla. So on the y-axis here, we have relative abundance, and on the x-axis here, we have our individual pigs. So over here in our left cluster, we have our limit-fed organisms, so these are our lean organisms. And then over here on the right, we have our ad libitum organisms, or our obese pigs. So first thing that kind of jumped out to us was this decrease in Firmicutes here, and then an increase in Bacteroidetes, as well as an increase in Spirochetes. And again, you might be thinking, Haley, what the heck are spirochetes doing in a gut microbiome? Because they're not really what we think of as a gut constituent. And actually, spirochetes are quite common in pig microbiomes, as well as other non-model organisms, such as the giant panda. And they have a lot of cellulose-degrading enzymes, so we believe that that's kind of their purpose, but we're not really sure why they're an important part of the gut microbiome. So phyla can give us kind of a good overview, but to really get down to the nitty-gritty, we can look at the level of species. And again, this is set up the same exact way. It's kind of messy, so I'm going to zoom you in a little bit. So the first major difference here was a significant increase in Treponema porcinum, which belongs to the phyla Spirochetes. Significantly increased in our ad libitum animals as compared to our limit-fed. We saw a significant increase in Prevotella species P592 in our ad libitum animals as compared to our limit-fed, as well as a decrease in Lactobacillus amylovorus, which is quite interesting because recently it has been connected to the amelioration of obesity. So in conclusion, for our first study, our fully developed ad libitum pig cohort seemed to display the metabolic characteristics of obesity similar to those of humans, and we observed distinct microbiomes between the lean and obese pigs. And specifically here I'm talking about Treponema, Prevotella, and a decrease in beneficial microbes. So now that we kind of had the endpoint characterized, we were really interested in kind of digging into the questions. So with the second part of this study, I was interested in looking at how the microbiome develops into these consortia that we see at the endpoint of obesity in our model. So really kind of probing that time component. So to do that, I took 12 post-weaned Megalitza piglets, so they were very small, woolly, and cute. And we divided them up into 3 groups. So you should notice the 2 returning players here. We had our ad libitum group that was given unlimited access to the basal feed, and our limit-fed group that again was given 40% of the caloric intake of our pigs, as well as a third group, which is an ad libitum group, so unlimited access to food, but their food had a high-fat component added to the diet. So we were interested to see if adding that high fat might kind of expedite changes within these piglets towards the endpoint of obesity in our model. So all of our piglets were kept on their respective diets for 18 weeks, and we collected weights weekly, back fat measurements by ultrasound biweekly, and periodically collected feces through fresh catch collection. And if you are interested in how that went down with pigs, please ask me at the end. It was quite interesting. Okay, so first let's get into the phenotypic data. So as you would expect, of growing organisms and growing piglets. All of our groups gained weight over time. So on the x-axis here we have weeks post-diet change. On the y-axis we have body mass in pounds. And we see kind of all of our groups increase in weight, which we expected. However, starting in about 8 to 9 weeks post-diet change, we saw the ad libitum and high-fat groups kind of significantly increase in body mass as compared to our limit-fed animals. And even further, starting kind of towards the end of the experiment, we noticed a separation between the high-fat groups and the other two, where the high-fat animals had a significantly higher body mass as compared to our other two groups. And when we looked to see where that was coming from, we noticed starting very early, about 5 weeks post-diet change, that the ad libitum and high-fat groups had a significant increase in fat deposition as compared to our limit-fed. And even further, as we moved along during the experiment, about 14, 16 weeks post-diet change, we noticed that there was a significant increase in fat deposition in our high-fat animals as compared to both the ad libitum and limit-fed. So really that high-fat component to the diet was driving that increase in body mass, and that was due to an increase in fat deposition, which is great. It's what we wanted. All right, so let's get into the microbiome data. And again, I know this is very messy, so I'm going to zoom you in and highlight some of the significant changes that we observed. All right, so this is set up pretty much the same as my other relative abundance plots. So on the y-axis here, we have relative abundance. On the x-axis here, we have our our 3 groups. So in this left section here, we have our limit-fed organisms from day 0, and then moving left to right in time towards 18 weeks post-diet change. Our ad libitum group here from day 0 to 18 weeks post-diet change, and our high-fat group from day 0 to 18 weeks post-diet change. And interestingly, we saw Treponema porcinum kind of pop out again as being significantly increased in our high-fat organisms as compared to our ad libitum and limit-fed. Interestingly, we saw a decrease in Prevotella within our high-fat animals as compared to our other 2 groups, so kind of contrasting what we observed in our endpoint. And then we saw another returning player, Lactobacillus amylovorus, and he was significantly, decreased within our high-fat animals and our ad libitum animals kind of towards the end of our experiment as compared to our limit-fed groups. And finally, though he be small in abundance, we saw a depletion of Streptococcus hydrointestinalis within our high-fat group starting at about 10 weeks post-diet change. So keep that in mind. And then when we looked at kind of the correlation between our phenotypic data and our microbial species, we noticed some interesting correlations. So just to kind of orient you to this plot, this is representative of correlations that have an absolute value of 0.6. So that was kind of our cutoff for a strong correlation. If you see a red circle, it means it is positively correlated. There are 2 things, so say like back fat and this bifidobacterium, they're positively correlated with each other. And if you see a blue circle here, that is representing a negative correlation. So the 2 that really popped out to me, again, was this Treponema porcinum. He was very highly positively correlated with both back fat and weight gain, so kind of what we want to see. And then ones that weren't maybe so significantly different within our kind of relative abundance and differential analysis plots, we did see some kind of beneficial microbes, such as this Bifidobacterium thermophilum, be negatively correlated with back fat and weight gain. So some things are lining up and correlating appropriately. So one of the many advantages of using shotgun metagenomic sequencing was the ability to look at viral populations. So this is a relative abundance plot set up kind of the same way, looking at viral genera. And what should immediately pop out to you is kind of the rapid depletion of this very large gray bar here within our high-fat group starting at 1 week post-diet change. And those correlate to SFI21DT1 viruses. And interestingly enough, they are Streptococcus targeting species. And so there are multiple strep targeting species that we saw differentially abundant within our high-fat animals. So I'm going to kind of zoom you in and let you take a look. So first of all, we have this Streptococcus virus, SFI21. Again, he's rapidly depleted here at 1 week post-diet change. I think the most convincing one is Streptococcus virus by ABC2. Starting at about 1 week post-diet change, we have a little bit of a blip here, but relatively compared to where they started, they were pretty depleted within our high-fat animals as compared to our other two groups. And then finally, it's not very contrasting up on the screen, but Streptococcus virus 7201 was rapidly depleted from our high-fat animals as compared to the other two. So really, we saw a ton of kind of rapid restructuring of viral populations within our high-fat animals. And interestingly enough, if you remember back to a couple slides ago, we didn't really see Streptococcus deplete from the gut until about 10 weeks post-diet change. So we're seeing these kind of rapid changes in bacteriophage targeting bacteria that are still within the gut, but they are being depleted. So overall, the microbiome of young pigs seemed to be kind of resilient to diet perturbation, and even at 18 weeks, changes in bacterial composition seem to be modest. So this might indicate that changes at the endpoint that we see at the endpoint of obesity might occur more slowly than we expected within our pigs. However, with the addition of high fat to the diet, this seemed to kind of expedite shifts in microbial compositions that are later observed in the fully developed obese phenotype seen in the Manglitza pig. And specifically here, I'm talking about the increase in treponema and the decrease in beneficial microbes. And we noticed that bacterial populations associated with an obese or lean phenotype seem to correlate appropriately with that phenotype. And finally, while bacterial populations didn't show drastic changes, phage population seems to be much more sensitive to diet change, and specifically here, I'm talking about Streptococcus targeting phage. And just in general, I didn't highlight some of the nuances that we talk about in our published paper, but in general, we saw a lot of restructuring of bacteriophage within the microbiome that occurred a lot more rapidly as compared to bacterial populations, which kind of points that something is going on within the gut environment and phage. And though this gave me a lot of really good information, it didn't really let me dig into a certain mechanism. So for the next part of my dissertation, I was really interested in determining what mechanism is driving changes in phage populations during the development of obesity. And specifically, I wanted to focus on immune activation. So when thinking about how to kind of set this study up, really I wanted to focus on how immune activation might influence bacteriophage reproduction, because that in general can have really impactful contributions to the gut microbiome. So just to kind of remind you, we have the lytic cycle over here, the lysogenic cycle over here, where they can integrate within its host. So kind of thinking about the steps within these and kind of what I wanted to focus on, the first thing that kind of popped into my mind was absorption, because if immune activation can influence absorption, again, that could be really, really impactful for the amount of virions produced through an active infection. And further, I kind of wanted to look at the endpoint of these reproductive cycles and progeny production. And with lytic phage, I focused specifically on just kind of progeny production, so how many virions are produced over time just from a virulent phage infection. But with temperate phage, you remember they can undergo lysogeny, so that gave me a really unique opportunity to look at how immune activation might influence prophage induction. So with my third chapter, I investigated just that, which is titled The Effects of Reactive Oxygen in Chlorine Species on the Reproductive Capability of a Temperate and Virulent E. coli Targeting Bacteriophage. And this chapter was co-first authored by myself and Dr. Higgins. So to do this, we utilized two bacteriophage species that both targeted E. coli, so one virulent and one temperate. For our temperate phage, we actually isolated this Protoviridae from a porcine fecal sample, and we have not sequenced it yet, so we have initially named it PF2. And the cool thing about this phage is that it is intestinally adapted, so it will give us kind of a good insight into how phage that reside in the gut might be influenced by immune activation and immune stressors. And then for our temperate phage, we utilized bacteriophage lambda. And specifically, I utilized the lambda LZ613 strain, which was generously given to us by the Zhang lab in Texas A&M. So this strain came with a lot of really cool tools for me to utilize to really study prophage induction. So first, it is a temperature-sensitive mutant. And what I mean by that is depending on what temperature I grow this phage at, I can kind of dictate what life cycle it undergoes. So if I grow it at a higher temperature, it kind of forces this phage to be strictly lytic. However, if I grow it at a lower temperature, I give it the decision to freely decide whether it wants to undergo lysogeny or actively replicate through the lytic cycle. And in addition, when it integrates into its host as a prophage, it confers antibiotic resistance, specifically to kanamycin. So this was really, really handy when I was trying to make lysogens or prophage-containing bacteria to study prophage induction. And it allowed me to kind of make as pure of a lysogen culture as I could. Okay, so just to remind you, the two kind of steps in the life cycle I looked at in this chapter is adsorption. So how do immune stressors influence absorption rates, and also how immune stressors influence progeny production. And to mimic immune stressors that phage might encounter in the gut, I decided to utilize hydrogen peroxide, which is a reactive oxygen species, and hypochlorous acid, which is a reactive chlorine species, that are both produced by neutrophils during the inflammatory response in the intestine. So my two big questions here: how do these reactive oxygen and chlorine species affect absorption rate, and how do they affect progeny production? So first, I'm going to start with the data for PF2. So first, we looked at the absorption kinetics for PF2 and how they are impacted by our immune stressors. So to do that, we took our sensitive E. coli host. We co-cultured them with bacteriophage PF2 in the presence or absence of our stress-inducing agents, so hydrogen peroxide or hypochlorous acid. We removed a sample at time 0 and then every 2.5 minutes for 7.5 minutes. We filtered the sample through a 0.2 micron filter, and this allowed us to remove all bacteria as well as phage attached to the bacteria that are actively adsorbing. So we are just measuring free phage within the culture. And from that, we plated it using a double auger overlay method to enumerate the plaque-forming units per mil, or the PFUs per mil, and then we plotted this over time. And unfortunately, we can't really measure adsorption directly, so we can measure it indirectly by looking at the loss of free phage over time. So as we move throughout our experiment, we expect to see a drop in free phage as they adsorb to their host and are subsequently filtered out. All right, so with hydrogen peroxide, we saw kind of a dose-dependent inhibition of adsorption. So to kind of orient you, on the x-axis here we have time, on the y-axis here we have relative PFUs. Our blue line here is going to be our control, and our red line here is going to be our treatment. So at 0.5 millimole hydrogen peroxide, we didn't really see a difference, but at 1 millimole and further 2 millimole, we saw an increase in this inhibition of adsorption. So it took a longer time for PF2 to adsorb to its host. And the story was the same with hypochlorous acid, but hypochlorous acid is much more toxic, so we had to use lower levels. So at 0.005 millimole, we didn't really see any differences here in adsorption rates. However, at 0.008 and further 0.01 millimole, we saw again this dose-dependent inhibition of adsorption rates. So to look at progeny production, we utilized a one-step growth curve. And before I kind of get into this data, I want to kind of describe a general reproductive growth curve of a virus or a bacteriophage. At this point here, this is where the virus is added. Time is going to be on the x, and y-axis here is kind of the relative virus count you can think of as in the culture, right? So when we initially add the virus, we will have that initial concentration. They will undergo absorption and injecting the genetic material into its host. And we have this kind of latent period where we see no growth in virions. And that's when kind of we're replicating the genome, we're transcribing, translating proteins. And once we start to assemble and release, we really see this logarithmic increase in virion concentration within the culture. And the amount of virus that is released within this log growth is called the burst size. Okay, so this is kind of a burst event. This is the measurement of how many are produced is the burst size. So, like I said, to do this, we used a modified one-step growth curve, and I kind of have a simplified model here for you. So we had co-cultures of E. coli and PF2. We incubated them for 10 minutes to allow for absorption because, again, we looked at absorption before. And then after the 10 minutes was up, we diluted them 1 to 10 in LB broth and added our immune stressor or a water control. We allowed them to incubate at 37 degrees for 60 minutes, and then every 5 minutes during the experiment, we took a sample and plated them to enumerate the plaque-forming units per mil. So first I want to take a look at the hydrogen peroxide data. So just to orient you here, on the x-axis we have time in minutes. On the y-axis here we have plaque-forming units per mil. And the blue line here is our control. So we can again kind of see this latent period burst. Latent period burst. And this kind of growth pattern is what I like to call a stepwise growth pattern, where we have periods of latency and growth, and latency and growth. And that's kind of typical of what we saw with PF2. However, when we add our lowest dose of hydrogen peroxide, so 0.5 millimole, we see a delay in this burst, so for about 5 minutes. But we do see kind of this stepwise pattern of growth, but becoming significantly decreased here towards the end time points. However, when we added 1 and subsequently 2 millimoles hydrogen peroxide, we kind of lose that growth pattern altogether, really becoming significantly decreased again by the end of the experiment. And when we look at the actual burst size, again, we see that 1 millimole and 2 millimole hydrogen peroxide was severely reduced. Burst size was severely reduced as compared to our control. However, when we take a look at hypochlorous acid, we really didn't see that much of a change. So again, x-axis we have minutes, y-axis we have PFUs per mil, and our blue line here again is our control. We see again that kind of stepwise pattern, and when we add our lowest dose, so 0.008 millimole hypochlorous acid, we don't really see any differences between that treatment and the control. However, when we add our highest dose of hydrogen peroxide, we don't really see any kind of indication of differences in growth until the very last time point, which might indicate it takes hypochlorous acid a longer time to have an effect on the host in terms of virion production. And as you would expect, there was really no differences between the burst size of our treatments and our control. So I have this little pictorial to kind of sum up the results for this part of the study. So all of the arrows here represent some length of time. So in our control, if we have our host, our host E. coli and our bacteriophage PF2, it takes some amount of time for them to adsorb to their host. Here they're actively replicating. I know it's not really the virion goes in, but it's a good little pictorial. And then we have some length of time to where it subsequently bursts out of its host. When we add hydrogen peroxide, it takes a longer time for PF2 to adsorb to its host. And when we add hydrogen peroxide again during this kind of like latency phase, we see a longer time for the burst to occur, as well as a decrease in virions produced. In contrast, when we added hypochlorous acid, we did see this elongation or slowing of adsorption to its host. However, when we added hypochlorous acid to the actively replicating PF2, we really didn't see any differences in virion production except for that very last time point at the highest concentration. All right, so now I'd like to get into the data for lambda. So again, we performed an absorption curve, and again, remember, I'm measuring the loss of free phage over time here. So with hydrogen peroxide at 1, 2, and even 3 millimoles, we saw absolutely no difference between the control and treatment in terms of absorption rate. So again, x-axis here we have time, y-axis we have relative PFUs, and even in our 1 millimole, like you can't even see the red line because they are so closely on top of each other. Yeah, so there was absolutely no difference between our treatment and controls here, and we even upped it to about 3 millimole, which was really stressing the bacteria out. And again, no differences in those. And further with hypochlorous acid, the same story was true, where we saw absolutely no difference in absorption rates between our treatments and our controls. So finally, my probably most fun project in this part of my dissertation, I developed kind of a prophage activation assay in which I grew up pure cultures of my lambda LZ613 lysogen. Remember, when this strain integrates as a prophage, it confers antibiotic resistance to kanamycin. So I grew it in kanamycin-containing media to an OD600 of 0.1. I centrifuged it down and reconstituted the lysogen in fresh LB broth. I aliquoted them out into separate flasks and either added a treatment or a water control, or PBS control, sorry. And then from this, I either collected a filtered sample every 60 minutes for 6 hours to determine kind of the virion count over time, and to look at how kind of the culture was affected overall. I obtained the OD600 every 20 minutes for 12 hours. So first, let's take a look at the hydrogen peroxide. So on the x-axis here, we have time in minutes, and on the y-axis here, we have the OD600. So here's our control line here in blue, and as you can see, it very quickly enters log phase, and we have this really nice pattern of growth. However, as we add hydrogen peroxide, we see dose-dependent delay in log phase. And you'll have to trust me when I say all of these here are significant, as well as here. I just didn't want to kind of overload the slide with a bunch of asterisks, but they definitely are. And even really 3 millimole as compared to kind of the sensitive host alone. The sensitive host alone had a delay in log phase for about an hour and a half. When we added hydrogen peroxide to the lysogen, however, that pushed the delay in log phase to about 3 hours. So really indicating maybe it's just more than hydrogen peroxide stress causing a delay in growth. So when we took a look at kind of the changes in plaque forming unit counts in the culture over time, we did notice that 3 millimole was significantly higher. So just to kind of orient you, on the y-axis here we have time in hours. On the x-axis here we have fold change as compared to our initial time. So with the control line here in blue, we kind of had this like sustaining amount of PFUs, but rapidly a decline in kind of baseline down here around 3 hours. And we see again kind of this dose-dependent effect of 1, 2, and 3 millimole, where each of them kind of have this kind of delay in reaching baseline as compared to the control. So 1 millimole kind of reached levels of control at about 3 hours, 2 millimole about 4 or 5 hours, and then our 3 millimole was actually significantly higher as compared to our control at 2 and 3 hours, and didn't really reach baseline until about 6 hours. All right, so then looking at our hypochlorous acid, so it kind of had the flip effect. So rather than kind of affecting log phase here, we saw kind of this significant decrease in growth starting at about 4 hours and continuing for the rest of the experiment. And there really was no difference between treatments. It was just all kind of treatments were significantly lower as compared to the control. Interestingly though, we didn't see any differences in plaque forming units per mil as compared to the control. So here again in blue, we have a little bit of a spike in our control, but it rapidly decreases in kind of baselines. And we see that pattern for all of our other treatments where they kind of increase, and if you look hard enough, the lines are a little stacked on each other in a dose-dependent manner, but definitely not significant. And again, they all rapidly decrease in kind of baseline to controls. So to sum this up, we have adsorption over here, prophage induction over here. So in our control, again, it takes some amount of time for lambda to adsorb to its host. And we don't really have any in prophage induction within our control. We might have a little bit of leakiness, but nothing really profound. But when we add hydrogen peroxide, we don't see any differences in adsorption. However, we see this prophage induction with a lot of virions produced by the treatment. And finally, with hypochlorous acid, again, we don't really see any differences in adsorption, and unfortunately, we didn't really see any differences in prophage induction. So to kind of sum my dissertation up, I was able to kind of determine how bacteriophage and bacteria are affected during the development of obesity and investigate a potential mechanism by which this might occur. So during the development of obesity in the Manglids of pig, it led to rapid shifts in intestinal bacteriophage populations. And I found that immune stressors differentially affected 2 E. coli targeting phage, highlighting the potential of immune activation to alter intestinal phage populations, but in different ways. So I have kind of a graphical abstract of the gut, and I know this is kind of busy, so I'll walk you through it a little bit. So on the left side here, we kind of have our steady state microbiome, where we have a good presence of the immune system within the gut. We have a healthy consortia of microbes. You can see our little friend spirochete there, some streptococcus. And we have a healthy consortia here of bacteriophage actively replicating, not inhibited in any way. However, in the obese microbiome, we have this kind of low-grade immune activation. Here's our neutrophils producing reactive oxygen and chlorine species. Really kind of with the microbes being altered, so you can see an increase here in spirochetes, a decrease in streptococcus, and really we see prophage induction, changes in bacteriophage composition. So overall, really kind of tying together my studies. So I'd like to acknowledge a ton of people for their help throughout my graduate career. First, my advisor, Dr. Elizabeth Schwartz. I could literally have not done this without her. Most amazing advisor. I could talk for hours about how amazing she was. To my committee, Dr. Lyles, Dr. Mary Medanza, and Dr. Michael Green. You guys have been invaluable in the development of my project and really pushing me to get this research done. To my outside leader, Dr. Stuart Price, for taking the time to read my dissertation and really give his really wise input, especially on the phage project. The Schwartz Lab, oh boy. First, Peter Rogers, I would like to acknowledge he was one of the main people to really inspire me for all of these graphics and really help me make them in my presentation. Adesola, Robert, Kia were all really big assets in getting this research done. And then to our new grad students, Sam, Aaron, and Faith, really keeping the happy environment alive in the lab right now, and the countless of undergrad students that have helped me with all of my projects. I'd like to thank my funding, the Hatch Grant, and the internal grant program, and my friends and family, my parents, my grandparents, my sisters, my partner Kyle, and many, many different friends that I have met along the way. And with that, I will take any questions. Thank you. Oh, bye.
Inflammation in the woolly pigs, either on Adelaide diet or normal diet or high fat diet.
Yeah.
Did you show us the inflammation that increased?
So I didn't, and back then when the pigs were alive, I know. Back then when the pigs were alive, there were not a lot of analytical tools to measure inflammatory markers, especially in woolly pigs. So we tried to measure specifically like lipocalin-2, but unfortunately we didn't really see any differences. So it might have been other different inflammatory markers that we didn't measure, or maybe it just takes longer for that inflammation to occur in the specific model.
What about resident neutrophils?
We did not measure those. Oh, Dr. Lyles?
Yeah, so excellent talk, Haley.
Thank you.
I was really intrigued by the observation you saw the Streptococcus phage change more rapidly than the Streptococcus bacterial population. And it got me thinking about the dynamics of that, and that thinking about Streptococcus DNA that would probably take a longer time for Streptococcus DNA to change, right?
Mhm.
But I think your data might indicate a change in metabolic activity in the Streptococcus population.
Mhm.
And so, would you regard your Streptococcus phage changes as indicative of a reduced metabolism in the Streptococcus bacterial populations?
That's a really good question. I typically kind of thought about it as kind of either a rapid changes in gene expression or maybe the addition of like the high fat component kind of, changed the metabolism of the Streptococcus bacteria. Kind of in some way, those kind of interplaying and not allowing these strep phages to actively replicate with their host.
Just a question for you, is it possible that changes in their metabolic output or profile would affect their ability to be infected? I mean, would it change
Perhaps both of
susceptibility?
in the phage receptors, but also
Yeah.
if there's a virulent phage in the Streptococcus, the host metabolic activity can be a big factor. Phage
Just whether it's
Yeah.
okay.
Wait, is 1 millimolar hydrogen peroxide, is that a physiologically relevant concentration?
That's a really good point. So it's around the amount at which it's like the highest kind of dose that neutrophils, around the highest dose that neutrophils can kind of take. I actually looked this up a couple days ago because I knew I was going to get this question. It's around kind of the lethal, like the highest dose before it becomes lethal to neutrophils. So in theory, it could be physiologically relevant. I scoured the internet and I'm not sure if people have actually measured like how total populations of how much hydrogen peroxide and hypochlorous acid is like released during like a respiratory burst or in an inflammatory event, but I can tell you doses higher than like 10-20 millimole will kill neutrophils. Yes,
And bacteria.
and definitely bacteria. Definitely bacteria. Yes.
I want to ask you a question about abundance plots.
Yeah.
What normalizes in the abundance plot, and how do you normalize out the problem of all of a sudden have more? Of,
Sure.
yeah, so how's it normalized?
Yeah, so I used Metaflan 3 to annotate my sequences, and they have their own kind of normalization method, so I didn't normalize them myself. From what I understand, they don't really explain it super well in their documentation, but from what I understand, they take kind of like the raw hits and look at the genome size and the genome count, do an equation with those together, and give us the relative abundance.
Do you see overall more bacteria in fat pigs?
Ooh, not really. It kind of stays the same level. I didn't see any like significant increases in either phage or bacterial populations.
So how do you measure that?
Looking at the total sequence count, the depth, and just like how many bacteria.
Is there a more quantitative way to, I mean, if you took a per gram amount and just colony counts, I
Yeah,
guess is some,
sure.
yeah, that's
But-
sort of crude compared to getting everything, but
Yeah.
I mean, if you had one bacteria, then you could monitor it.
Yeah, definitely you could do
You
that.
just thinking more per gram?
I was wondering, like you just said, if you have one thing that's now super abundant, and so
Yeah.
you get lots more reads for that. So then all of a sudden the abundance of something is 10%, but the raw number was that same, right? So if you had one organism that you thought was constant, you could look and say, I always have the same number of reads
Uh-huh,
for that organism, and I could say,
yeah,
relative
compare
to
everything
that.
else to
Yeah.
that.
But it is hard to normalize your data, right?
Yeah, it's definitely hard.
You would have a consistent population of anything,
Yeah,
but and CFU counts would also be biased, right?
exactly, to aerobic
Yeah.
bacteria or
Yeah.
aerobes.
So, I mean, your total biomass, I think that's what you're kind of getting at, is there a change in actual
Yeah.
numbers of cells, right? By thinking about fluorescence. But the actual data you might have is DNA yield.
Yes. Yes.
So what does that tell you?
What does DNA yield tell me?
Maybe we'll explore that in the,
Yeah,
in
we
the
can
future questions.
Stay tuned.
Have you looked at ROS and RCS together? I assume
I
those events
haven't.
happen independently within the organism?
Yeah.
Or those constant stresses?
So they're kind of like they're definitely attached within the respiratory burst kind of pathway within neutrophils, and so technically they would kind of be released at the same time. And I didn't look at that, but that was definitely like on my laundry list of things that I wanted to do.
Additive, synergistic?
Yes, definitely. I think it would be synergistic.
I do have one more question,
Yeah.
which is about phage absorption
Yeah.
of dead cells.
Dead cells.
Yeah. When the cells are dead,
Mm-hmm.
do they absorb phage or no?
Oh, dead in what way? Like kind of like
Dead as in
like have they like?
Let me parse that question.
Yeah, I'm gonna specify before I answer.
Back a little bit.
But,
Did in what way?
I mean, dead.
So I mean like broken up into pieces, membrane still
No, I'd
intact.
say, okay, let's say, treating with an antibiotic that arrests their growth.
Sure.
And-
So
And maybe even like a bactericidal one, but do you
Yeah.
still get absorption at the normal rate? Or the cells need to be alive to absorb phage?
so I think it depends on a lot of different factors. I think it depends on the receptor. So for example, lambda's receptor is a maltose porin, basically. So if it is not metabolically active, if it is dead, it's probably not going to be expressing that, therefore it cannot get in. If it's a more ubiquitous one like LPS, I would imagine it could absorb, but it's not really going to be able to do anything after that. So I think it depends on the receptor and the bacteria in general.
How would you test it?
Oh, I could apply a bactericidal antibiotic to the culture and measure absorption over time.
But you didn't do that.
I didn't do that.
Because you only have, basically, you don't, on the plot that you showed us, I think you said it, but I
Yeah.
don't think I saw it, where you don't have any treatment and any phage. What's the growth rate of the E. coli before you ever?
Oh,
So does it
yes.
slow down at all from getting the phage or getting any?
Yeah,
growth
so
treatment?
so we ran a bunch of growth curves to determine kind of like concentrations of both at which it stresses the bacteria out, but they're still able to grow. So they should. I didn't use concentrations in which they should be dead, but I also didn't use concentrations in which no.
It stresses the bacteria out, that means.
So it activates stress response in terms of DNA damage, hydrogen peroxide damage in general, that kind of stuff. Yeah, slows their
That's
growth.
what you
Yes, that's what I mean. Sorry, lots of stuff going on up here right now. Thank you guys. Thank you, everyone.
All right, committee members, if we could reconvene in 5 minutes back here. Or maybe, if you need 10, that's fine too. Here, you can come through.
Okay, I will disconnect you.
No.
Good job.
Thank you. Thank you so much.
How well?
Yeah.
Is that good?
Very, very good. Very good. I hope you feel proud.
I do. I feel really good about
What I
that.
really appreciate is just how well you clarified what you were talking about. So, really, really
Thank you.
couldn't really expect better.
I just want to say congratulations, Haley. It was
Thank
really
you.
good. I really
Thank
enjoyed it. Just like I really enjoyed Kia's when she did hers.